Good morning, everyone. Thank you very much for having me here today. If you're not from Vancouver, I wish I could tell you that the weather gets nicer than this, but it really doesn't. So just make do with what you get. I'll be talking to you today about the research that I was fortunate enough to conduct over this past summer of 2012. As you can see from the slide, I was working at the Vancouver campus of the University of British Columbia with the generous help of Dr. Nick Famelli. So without further ado, let's embark on the exciting journey that is stochastic modeling of calcium transport in cytoplasmic nanospaces of vascular smooth muscle cells, or we'll go with VSMC for short today. So what you see here on the screen is a crude representation of Dr. Sidney Ringer's experiment performed back in 1883. While working with amphibian hearts, what Dr. Ringer did was his less than painstaking experimental procedures led him to suspend his amphibian heart in a solution of pure London tap water, all the beautifully clean London tap water that we all know and love. Obviously, this water would be considered hard water, meaning there were significant amounts of ions like potassium, sodium, magnesium, and for our purposes, most importantly, calcium ions. This heart, in particular, maintained healthy, high-pressure contractions for several hours, so nothing new. He saw what he expected to see, and what came next is the interesting part. So one day, purely by chance, Dr. Ringer replaced that tap water with a vat of distilled water, and what he saw, to his surprise, was that the heart was only able to beat for a couple of minutes, and it automatically died out. What he didn't understand then, but what we understand now, is that calcium ions play a massive role in not just heart contractions, but in a lot of physiological cellular processes. Before I continue any further, I'd just like to briefly clarify some anatomical details regarding smooth muscle in general. What we have in this blow-up illustration is a sort of cartoon representation of a typical vascular smooth muscle cell. Vascular obviously meaning blood vessels, and smooth muscle as opposed to cardiac or skeletal. And the main thing to note here in this diagram is the red ring, if you will, of vascular smooth muscle cells that encase the diameter of our blood vessel. And pretty much it's the action of their actin and myosin filaments, their contractions that causes the blood vessels to contract, pump blood, and allow us to go about our daily lives. So how exactly do calcium ions work, they're wonder, in these vascular smooth muscle cells? Well, the short answer to that would be wave-like oscillations. And that's in the sense that the concentration of calcium inside these cells, they display a periodic pulsing between a low resting state and a high contractile state. The traces here were obtained by Inu et al., obviously, in 1994 while stimulating rat tail arteries with electrical impulses not too unlike neuronal impulses. And what they found using confocal fluorescence microscopy, as you can see on the top row there, is that, well, point A on the graph is where the shock is administered. So point A would be our impulse in this case. And they saw that at each location where they were measuring, they saw calcium concentration in a single cell oscillating up and down in a uniform method, which obviously brings up the question, why the oscillations? You would expect, someone new to the topic would expect that the calcium concentration might increase significantly for a little bit of time and die out as time goes by. Well, it turns out the areas of close association between the plasma membrane and the sarcoplasmic reticulum are otherwise known in our lab and around the world as PMSR junctions. They're crucial in maintaining those calcium oscillations. Comprising about 15% of the cellular diameter, the cube nanometer scale volumes between the plasma membrane and the SR, they actually harbor handfuls of calcium ions at a given time. Now, these calcium ions are not enough in numbers for bulk diffusion equations to be applied. So we need a way to analyze and track these calcium ions individually. And And the techniques for that will be discussed just a little bit later, but for now, let's just look at the role of PMSR junctions in a little bit more detail. So the iconic experiment that shows sort of the importance of nanospace junctions in vascular smooth muscle cells was the Killiculin A experiment. That was actually performed on the UBC campus with Dr. Van Bremen's laboratory in the Department of Anesthesiology. And what we saw was that without exposure to Killiculin A, the control cells display the beautiful wave-like oscillations in calcium concentration. In vitro, this was done. But with increasing doses of Killiculin A, the control cells displayed, the non-control cells displayed less and less noticeable wave-like oscillations until the sort of threshold Killiculin A concentration where the wave pretty much just died out. We should note here on the graph that the sarcoplasmic reticulum, short here for SR, it actually does retain its function. It's just the oscillations that follow after that initial impulse that straight away died out. So this is interesting, and it pretty much equips us with all of the biological background that we need to understand PMSR junctions. Rough background, I would admit, but we'll just move on for now. Coming back to that topic of analyzing the calcium ions in the cytoplasmic nanospaces, we can ask these two questions. First being, how can we visualize that calcium transport processes within these nanospaces in real time? So in not just at a given moment in time where we can take a micrograph of the cell and look at it fixed in time, but in a more realistic process. And the second question would be, how can we log this corresponding quantitative data? The first part should be obvious to you by now that PMSR junctions in vascular smooth muscle cells are much too small to visualize by optical or confocal microscopy, which would give us realistic, real-time data, but that's not very useful for nanometer scale spaces such as this. And electron microscopy can give us beautiful images like this one to work off of, but all it can provide us is the cellular architecture. So what we did, Dr. Fameli and I in our lab this summer, was use a program called MCell to pretty much recreate the cytoplasmic nanospaces on the computer and run simulations with parameters that can be freely changed by that user. So in the case of our smooth muscle cell, we've created the plasma membrane containing calcium sources, which are in the literature referred to as NCX, sodium calcium exchangers, and the corresponding sarcoplasmic reticulum containing calcium sinks, or SERCA, or sarcoplasmic endoplasmic reticulum calcium acceptors. Oh. In the case of this vascular smooth muscle cell, the densities and the reaction kinetics, all the transporters were taken straight from the literature, so we can rest assured that the reactions going on in these simulations were scientifically accurate. And just touching on the data output, this is a very crude model of what the capture rate of calcium might look like if you, on the top line, cluster the calcium sources right on top of the calcium sinks, and on the bottom, if you randomly distribute them over the surfaces. This obviously is quite trivial if you think about it, but it does open the door for a lot of possibilities where we could explore these nanospaces in a lot of detail without having to sacrifice any living cells pretty much. So I'm just going to show you a little animation now of what MCell and its partner programs can do for you in terms of visualizing this nanoscale spaces in the cell and what kind of information it can give you. So you see here in red the plasma membrane. That white dot dancing around would be the calcium ion, and it is modeled perfectly by Brownian motion in the MCell program itself. So it is a Monte Carlo simulation, and it would follow, including the transporters, it would follow the rules of biological physics to the level of, the ions themselves. members of our lab are currently working on these three topics here. That movie that you see playing in front of you is the model that I was working on this summer of a nuclear imagination and the calcium transport and movement processes that could occur in the inside the imagination itself. That's we're working on that, and we're looking to produce some papers on that within the next couple of years. Lysosome SR junction and mitochondrial SR junctions have already been modeled, and papers on that are in the works as well. So thank you very much for your kind attention today, and if I can take any questions, I guess this is the time.
Question from the floor?
Yes.
On one of your earlier slides, I'm going to have to go back to show me.
For sure, yeah.
About you'd expect the oscillations would just die out after. I wanted to clarify which oscillations are you pointing to?
Do you mean this slide by any chance?
No,
Keep going.
this slide here.
Okay, I guess I should have clarified a little bit about this black solid bar you see here. What that is the actual pharmacological implication of the impulse. And you can obviously see that with the start of that impulse, you can see a bit of a, if you just follow the colors, you can see the oscillations in calcium concentration pervading through until the impulse is stopped. Obviously, with that stop in the impulse, you would expect levels to just sort of diffuse out and die back down. Does that answer your question?
Yeah.
Okay.